Good morning and welcome. Today we're going to be looking at some genetic technologies, PCR, gel electrophoresis and DNA profiling. So we have PCR here, gel electrophoresis and 2 DNA profiles. To start off, let's have a look at PCR. So PCR stands for the polymerase chain reaction, and the whole purpose of it is to amplify DNA, which means to make more copies of it. Why we might like to do that is, for example, at a crime scene, we might only have a tiny section of DNA and we need to amplify it so that we have more to work with. So what we have here is a PCR machine, and these are the components that go into the PCR machine. And the process of PCR is we start off with the DNA and we heat it up to 95 degrees C. That denatures the DNA to provide us with one strand of DNA. And PCR is basically heating up, cooling down, heating up, cooling down. And we have these tiny PCR tubes here with a capacity of 0.2 microliters. And we use this micropipette here to put in all of the components into the PCR tube, which goes into the PCR machine. So let's have a look at the components. We have the DNA sample of interest. This might be, for example, from a crime scene. We have primers, which are short sections of DNA, and they basically start off the replication process. We have nucleotides, and these nucleotides will match up to the complementary strand to make a new DNA strand. And we have Taq polymerase. That's an enzyme that actually allows the nucleotides to bind. We also have a buffer, which maintains the pH of the solution. And it's really important to do that because the Taq polymerase is an enzyme that is sensitive to changes in pH, and the DNA itself is also sensitive to changes in pH. So PCR consists of, like I mentioned, heating up, cooling down, heating up, cooling down to make many copies. And an old-fashioned way of doing this was basically to have water baths in a lab of hot water and ice water, hot water and ice water. And you would have to physically move the tubes yourself. But now we have this thermal cycler that can do that for us. So now that we have many copies of our DNA, we want to cut it up. And basically to do this, we use a restriction enzyme. This EcoRI is one name of a restriction enzyme. Eco refers to the fact it's from E. coli, and RI refers to the strain. And basically, these restriction enzymes cut the DNA sections at a specific place, producing sticky ends. So this one here on the left produces ends that are not cut bluntly. It's cut at G, for example. It's important not just the letter, but also what letters it's next to. And it's cut at AG, for example. So it always cuts at a specific place. And you do get restriction enzymes that can cut straight down the DNA to produce blunt ends as well. So now that we have our sections of DNA, we can use gel electrophoresis to analyze our DNA. We have a matrix here, and we have 4 wells. On the well on the left, we might have ladder DNA, which is basically known DNA, and that is used for comparison. The next well, we might put in DNA from a crime scene, and the next 2 wells, DNA from 2 of the suspects. Now, the DNA is always negatively charged because the phosphate in DNA is negative, and it always moves to a positive electrode. And it moves according to its weight, how heavy it is. So the heavier the sections of DNA, the slower they'll be. And the smaller the sections will move much more quickly. So what do we need to have in the gel? Well, we need a dye. And the dye is also negatively charged and moves towards the positive end. And that shows us the movement of DNA. But it doesn't stain the DNA. It just shows us how far we have come in the process. And on the left is a diagram of a ladder of DNA that is of known DNA, and it can be known as our marker, and that allows comparison so that we can work out the weight or mass of our sections of DNA. So we'll carry this on in the next video, and I hope this has been helpful so far.